Good afternoon. For those of you online, my name is Lex van Geen. I'm the co-director of Columbia's Superfund Research Program, and I'm happy to introduce today our second speaker in the seminar. Louis Ziska is a plant physiologist by training. Received all this training I learned at UC Riverside. And has worked for different organizations, but the longest stint was for a couple of decades at the U.S. Department of Agriculture in Maryland. Louis has studied the impact of various aspects of climate change on plants and on crops, including CO2, so carbon dioxide, UV, not only on rice, but also wheat and soybeans. He is interested in the impact of climate change on food security, and is going to tell us today about the impact of climate on carbon dioxide and plant biological links to public health. Welcome. I should have indicated that Louis also, after spending a considerable amount of time at USDA, is now a faculty member of the Maryland School of Public Health.
Thank you very much, Lex. I appreciate that. I want to talk about plant biology, not in the normal context of what you have on your plate, although that's obviously important, but I want to try and look at it in the context of what it means with respect to public health. So, tongue-in-cheek, I've sort of these as what we call the good, what we will refer to as the bad, and what I will refer to as the oceans melting Greenland. So we want to try and give you a sense of all these interactions, but before we do that, we have to make sure that we more or less are all on the same page when it comes to what we mean by that important phrase of climate change. What does climate change mean? And it means different things to different people. So I'm going to begin with a pretty obvious observation. We know that when you take a source of carbon, whether that source of carbon is coal or natural gas or whatever, and you oxidize it, you burn it, carbon, oxygen, carbon dioxide. So carbon dioxide, more or less, 90% of it comes from fossil fuels, about 10% from land use change. Where does it go? Well, about 50% of it stays in the atmosphere, about 25% is reabsorbed into land, and about 25% dissolves into water with effects in terms of lowering the water pH. It's been going up very rapidly. If you go back in geological time for at least the last 800,000 years, it's stayed steady between 200 and 300 parts per million. Now this is a little bit old, it's over 410 parts per million currently. How much will it go in the future? Depends on which model you believe. There's various models. We'll call this green line the everyone hold hands and drive a Prius model, which is probably not going to happen. The dashed line, which is the nomenclature somewhat changed, representative concentration pathways, 8.5, 4.5, and so forth. This is the 8.5 line. Pretty much where we're on at the moment. So we know that we burn fossil fuels. We know that adds carbon dioxide to the atmosphere. And the next question, of course, is, so what? Why should I give a flying fig whether it's 200 or 300 or 500 or 600? What difference does it make? Well, a couple of reasons why you should be concerned. One is the effect we know of in terms of the physics. And that is when you look at all the various components of the atmosphere, there are two components that stand out. One is carbon dioxide, the other one is water vapor, humidity, if you will. And those are considered to be global warming gases. What does that mean, though? What does that mean? And the best analogy I can come up with is music. And bear with me for a moment. When I play, if I tune a guitar, where two strings of the guitar are at the same frequency, and I hit one string, what will the string next to it that's been tuned to the same frequency do? It'll vibrate, right? What if I'm a Methodist? Will it still vibrate? If I'm a Methodist doing it, will it still vibrate? What if I'm a Republican? Will it still vibrate? I just want to make sure the laws of physics are not, changed depending on religion or, okay, just checking. No, that's absolutely true. And what happens is that the bonds of water vapor and CO2 are such that they also resonate, but they don't resonate in the key of A for a guitar, they resonate in the key of infrared or heat. So whenever heat is absorbed by one of these, they basically take some of that energy and vibrate, they resonate. And we've known this for 150 years, it's not new science. So if there was no water, if there was no carbon dioxide, the average temperature on the Earth would be about minus 18 degrees Celsius. It would be very cold. So having these greenhouse gases is, of course, to our benefit in terms of biology. But the thing to keep in mind, I think, is that the temperature increase is not uniform. When we talk about areas warming up faster than others, why is that? What's going on there? And the best way to try and look at it is to look at it in the context of water vapor and CO2. So where on the Earth is the air warm and humid? That is to say, where is water vapor the principal greenhouse gas?
Tropics.
The tropics, right? Okay. So in terms of the rise in CO2, we're not seeing a great deal of change in the tropics per se. So that's less affected. Now let's flip that around. Where is the air dry? And therefore adding more CO2 will have a bigger effect. At the poles. Right? It's the whole psychic network thing, man. That's really cool. I like that. So at the poles, where else is the air dry? In the deserts. It can be dry up as you go up in altitude. It can be dry seasonally. Winters are typically drier than summers. So both spatially and temporally, that shift in water vapor has some bearing in terms of what spots on the Earth are warming up faster than others. Now I realize I've taken an entire semester's worth of physics and atmospheric chemistry and condensed it into a few minutes, so bear with me, but hopefully we do a sense of what we mean when we say climate change. But there's another aspect of this, and that has, plant biology consequences of temperature, right? Less temperature increase for water vapor is high, more precipitation, greater temperature increase with latitude or altitude, increased desertification, increased drought, rising sea levels from increased polar and glacial melt. All this can have an effect in terms of plant biology. But there's another effect, and that is a resource and plants need in order to grow. So we know plants are essential to life at the bottom of the food chain. And I'm sorry, this is very, very basic stuff, but what do plants need to grow? Sunlight, what else? They need water, they need nutrients, right? You can grow them, you don't have to grow them in the ground, you can grow them aeroponically, hydroponically. And of course, they need carbon dioxide. Now, about 95% of all plants lack optimal amounts of carbon dioxide. And here I am, throwing it leaps over bounds and, highest it's ever been in the last million years. So that change of and by itself is going to alter all of plant biology. Just as if I were to change the amount of water globally, or if I change the amount of light globally, if I change the amount of potassium globally, it's going to affect every living plant system. Well, that's good though, because I went to the ExxonMobil website and they showed me this nice picture of loblolly pine, and you can see, wow, look at loblolly pine. It grows more when I give it more CO2, and isn't that wonderful? That's great, and that's fabulous. Oddly enough, however, they left out other plants that also respond to carbon dioxide, like kudzu. Anybody from the South? No? All right. So there are also some biological consequences of this, right? It's a fundamental resource. We know that all plants are beneficial for human society, right? And when I change a resource, the one thing I know for certain, being an avid gardener, not all plant species are going to respond to that change. If I put twice as much fertilizer in my backyard, I can tell you with great assurance that not every plant species is going to equally respond in terms of growth. And it will also alter qualitative components of plant biology. Well, let's pick up on that last point, qualitative components of plant biology. Let's look at it from the point of view of a plant that poses a medicine that's used globally. And the plant's in relation to CO2 and malaria. We know that malaria is a ubiquitous scourge around the world. In 2017, over 400,000, primarily children under the age of 5, died in sub-Saharan Africa. And we know that when it comes to malaria, there are different medicines. One of the medicines is what are called ACTs, or artemisinin combination therapies. There's a semi-synthetic method, fermentation of artemisinin and photochemistry, and then the agricultural method where you cultivate Artemisia annua, which is the plant species that provides artemisinin, and then cultivation and extract that for ACTs. This is sweet annie, or Artemisia annua, common roadside weed. I've seen it actually on the Hudson Parkway. Produces this compound, artemisinin, with this unique peroxide bridge that we think is, in fact, deadly to the common carrier for malaria, Plasmodium. But it doesn't, and it kills the Plasmodium almost immediately, but it doesn't kill all of it. And so what happens is that short-acting drugs are added to the artemisinin, usually some kind of quinine or quinine derivative, so that you increase the longevity of the drug, so that it has a much more efficacious application. But let's look at it from the plant biology perspective. If plants respond to carbon dioxide, what is the recent change in CO2 done to artemisinin concentration? So myself and a colleague, Chunwu Zhu at the Chinese Academy of Sciences, looked at artemisinin concentration as a function of growing the artemisia under different CO2 values. And what we found was that the more carbon dioxide, the higher the carbon to nitrogen ratio, and the higher the amount of artemisia. So we saw this nice correlation that the more carbon dioxide you gave it, the more of this particular compound was produced. But we wanted to look at this in the context not of projected changes in carbon dioxide, but what has happened to recent changes. I can show you that graph, the Keeling curve, where basically carbon dioxide has gone up by 30 already. So what we did was to sample herbarium throughout China throughout the 20th century with samples going back. The oldest sample was 1909. And we looked at carbon and nitrogen ratios there to determine or to estimate what the artemisinin concentration is going to be. And if you looked at the herbarium samples and the estimated artemisinin beginning in 1900 and going forward, you essentially see something very similar to what we see in terms of the recent change of carbon dioxide. Since this was published a couple of years ago, and since that time, the Chinese government is investing more in greenhouses upping carbon dioxide concentration to grow their artemisinin to get more, or artemisia to get more artemisinin. So we thought that was pretty cool. So the good, common weed in North America, maybe an essential pharmacological resource to treat malaria in Africa. Recent increases are associated with an increase of a known antimalarial drug from this plant. What other plant-based medicines are responding? How are they being affected by the change in CO2? We don't have a good answer for that yet, but obviously for many cultures, they don't go to the corner Walgreens. They have a pretty good sense of what their local pharmacological sources might be. Now let's segue to the bad. Plant-based allergies. Those of you that suffer from seasonal allergies will appreciate this. Typically, they fall into sort of three categories. In the spring, it's primarily trees. In the summer, it's weeds and grasses. And in the fall, it's primarily ambrosia, ragweed. And so we began back in the late 1990s looking at a very simple question of what does CO2 do with respect to common ragweed growth and pollen production. And this is just us collecting some of the pollen from catkins. And what we found was that if you went from pre-ambient concentrations to current at the time, we got about a doubling in terms of per plant pollen production. If we went to 600 parts per million, which we'll almost certainly hit by the end of the current century, we went to 20.5 grams. In addition, Ben Singer, who was a med student at Northwestern at the time, did some really interesting work with the ELISA sampling for antigen ambient A1, which is the principal antigen that causes the allergic reaction, showing that it also increased significantly with more carbon dioxide. Cool. Wow, that's interesting. But it is a laboratory study. How do we scale it up? How do we get it in a way that's meaningful? Well, this is difficult. Those of you that might be familiar with carbon dioxide research may be familiar with a method called FACE, which is an acronym for Free Air CO2 Enrichment. And what they do is to provide elevated CO2 in an open air system, in a real field-based system. And this is a picture of the Loblolly Pine system at the Duke Forest. It's hard to, let's see if I have another picture here. It's sort of hard to scale this, but this is about 100 to 120 feet across, pushing additional elevated CO2 into here. They have a very sophisticated network, so if the wind comes from this direction, they can compensate by adding more CO2 in the other direction, dot, dot, dot. Their budget, I think, for CO2 on a yearly basis was over $1 million. And this little, you may not see this, but this little path here is actually a railroad line, because they had to use railroad tanks of carbon dioxide over 6 tons a day to elevate the CO2 within the forest, right? It was a DOE study, which in our acronyms, DOE stands for the Department of Everything, so lots of money. How do we do that for ragweed? Yeah. Especially considering, you know, my entire budget for doing research at the time was about $2,000. So I could only do maybe, you know, 20 seconds, and that wasn't really going to work. How do we get to the real world? How do we get to the real world? And it occurred to me in reading a biography about Charles Keeling, why did he start measuring in Hawaii? Of all the places he could have monitored carbon dioxide, why did he choose Hawaii? I mean, you know, admittedly they have, you know, great daiquiris and timeshares and all that good stuff, but why there? Why monitor carbon dioxide in Hawaii at 10,000 feet? Oh, because that's background carbon dioxide. That 410 part per million, trust me, it's not what the CO2 concentration is in this room. It's not what the CO2 concentration is outside on Haven Avenue. So what if I did that? Instead of using the most geologically or geographically isolated spot on Earth, what if I looked at ragweed in real time by using a natural urban-rural gradient? And that's what we did, because it was cheap. And I went to the City Fathers in Baltimore and said, will you let us do this? And so we set up a urban-rural gradient to study the initial growth, productivity, and species dynamics of fallow farm soil, which had ragweed in it, between a sort of organic farm to the suburbs to downtown Baltimore. Dig our plots, move our soil, same seed bank in the soil, made it uniform, monitored, and so forth. Now, as an academic, I have to show you at least one slide with a lot of numbers on it that nobody could possibly read. So this is my contribution. But basically, we did this at the beginning of the aughts and looked at daytime CO2, nighttime temperature, daytime air temperature, and the change in season length, the number of additional number of frost-free days in downtown Baltimore. And what we found was similar environmental characteristics consistent with what a near-term change in climate would be. So the CO2 was slightly higher, the temperature was slightly higher, higher nighttime than daytime, change in the season length. Now, obviously, we can't control all the parameters. One of the ones we were most concerned about was ozone. But what we found was that in comparing the downtown site to the farm site, was that as soon as you got a high ozone event in Baltimore, within a few hours, it was a high ozone event down at the farm. So we don't think that was a major difference. So all of this, we think, is fairly consistent with, as a harbinger, if you will, of what climate change might be. So we dug our sites in the Inner Harbor by the Science Center. You're familiar with that. If you look closely, you'll see Jimmy Hoffa's wrist watch. Sorry. So we put in our soil, we put in our rotorod samplers, and this is what we got, ragweed. And this is just to give you some sense here for the folks listening in. This is the farm site here, and this is the day of year when we first got pollen, and the pollen is being measured in grains per cubic meter. This is our maximum. Where'd you go there? Yeah, this is our maximum, and this is the day of year when we stopped getting pollen. And here's the suburbs, and these two lines are essentially correspond to this line here, the beginning at the control site, the farm site, and the maximum time here. So you can see that they're flowering, beginning to flower earlier. They're hitting their maximum before the farm site and then declining. But what really blew us away was what was happening in terms of the downtown urban site. And on a per plant basis, we're getting about 10-fold the amount of pollen in an urban site that we're getting in a country site. And this is in the Journal of Ag and Clinical Immunology. Yeah, that's nice, that's rural, that's great, but hey, it is a global problem. How do we scale up to the globe? How do we go to the continental level? That's very difficult. We can't exactly do a phase system that stretches from Canada to Louisiana. So what we did, what I did, is I called up a lot of allergists. And I said, hi, you don't know me, but I'm a plant person. And sometimes they'd hang up and sometimes they'd listen. And what we were able to do is to look at changes in seasonality as a function of latitude. Because remember me telling you about the CO2 and water vapor? That when you go to colder temperatures, you're going to have a bigger effect than when it's down when the humidity is high. Well, here's before going from East Texas up to southern Canada, and to look at ragweed data going back to the 1990s to move forward to see if we could pick up that signal in terms of the season length of ragweed. And so what we found wasn't a perfect correlation, but going from basically no change in the pollen season to almost a 4-week change when we got to southern Canada, beginning from the 1990s and going up to 2013. So this was a real signal in real time, taken from our laboratory observations and scaling up as we went. Still not satisfied with that, we wanted to do more. So now we went globally, and thank God for email and social media, and said, hey, you guys in Turku, Finland, what are you doing? And they actually had the longest continuous record of seasonal pollen. Now we're not just looking at ragweed, we're looking at what starts in the spring and what ends in fall. And this is going back to 1974, showing changes there. This is Reykjavik, showing changes in total seasonal pollen, but from the early 90s. Kansas City, Missouri, Geneva, Switzerland. And then this is a little hard to see, but basically what we did is to take, I think, 17 different locations in the Northern Hemisphere and to look at the percent change in pollen load per year as a function of the temperature increase, looking at the Tmax temperature increase and the Tmin temperature increase, and seeing essentially significant changes in the overall amount of pollen that were being experienced as a function of temperature for North America. So as temperatures warm, there is a definite effect with respect to pollen load. And just to sort of reiterate, when it comes to pollen, there's sort of three major aspects. One, of course, is the amount of pollen. The other one is the season length. And the third one is the allergenicity of the pollen itself. And that's a tougher one. I showed you some of Ben's work with the laboratory ragweed, but it comes much harder when you actually have to do it out in the real world and the plant's moving and the wind's blowing all the pollen everywhere and you're trying to capture the, no, not going to happen. Because you need, a fair amount of pollen in order to do the ELISA test. So here, in terms of the bad, rising CO2 and temperatures can influence the pollen season, the pollen amounts, and we're still working on pollen allergenicity. We don't have a good handle on that yet. Now we're going to get to the oh my God part. And we've been looking at the effects of rising CO2 with respect to potassium. And there's an inverse relationship between potassium and flammability. The more potassium you have, the less heat you put off, the less flammable you are. And one of the things we found when we looked at an invasive grassweed, Bromus cheatgrass, was that as we looked at recent and projected changes of CO2, the potassium concentration went down. So it became more flammable. We'd like to continue that work with wood to see if, in fact, as CO2 has risen and is projected to rise, what does that do for potassium levels in wood, and what does that mean for flammability of forests? And what impact will that have in terms of fires and air quality? Looking at ozone generation, kudzu, that nice weed I showed you earlier, produces VOCs, which are a precursor for ozone. If you give kudzu more CO2, it produces more VOCs. So what impact does that have in terms of ozone? Contact dermatitis. If you grow poison ivy under higher CO2 levels, it produces a more virulent form of erysiol. Narcotic intensification. If we have public health issues related to narcotic abuse, what were those narcotics, like erythroxylococa, be grown in the future as the climate changes? How will CO2 influence their ability to produce those chemicals? Anybody online right now?
Yes.
Would you type something on Google for me?
Sure.
Would you type marijuana and CO2? And then read me the first hit you get.
Maybe somebody can do it.
How do you use CO2 to increase yields in your marijuana growth?
How to use CO2 to increase yields in your marijuana growth. Golly, wonder what they know that we don't know. They don't publish in the scientific literature, though, do they? Shame. Food allergies. If we are looking at it from the point of view of difference in proteins, what does it do? Does it increase or decrease? I'd like to know. Food safety. As temperatures warm, what happens to post-harvest production? Are you going to increase the risk of E. coli, Clostridium, other food-based pathogens? What happens to pesticide use? Here's some work we did with Canada thistle, which is an invasive weed. We sprayed it with Roundup on the left, got pretty good control, but we sprayed it under ambient CO2. At 650 parts per million on the right, we sprayed it with glyphosate and got zero control. So how does CO2 affect pesticide usage? For now, let me just focus on one of these OMG issues, and that's nutrition. We have very strong evidence from a number of different laboratories that projected increases in carbon dioxide will reduce protein levels in a number of different crops. This is rice. We think that this change is not just for projected increases, but for also recent increases in CO2. This is some work that I was doing just prior to the government shutdown back in January, looking at 8 different lines of rice and looking at the change in protein from 300 to 400 parts per million CO2. That is the recent change. And I think 7 out of the 8 showed a significant decline. The problem was that after the shutdown, we lost equipment. We weren't able to repeat this, so this is only 2 replications, and I'm a little bit hesitant to publish it, but I don't mind sharing it with you to get a sense of what we're seeing. It's ubiquitous. Here's some work by Daniel Taub at the University of Texas, barley, rice, wheat, soy, potato, and the number of studies that were used to average that point are in the parentheses here. So 20 studies of barley, 14 studies of rice, wheat, and so forth. And you can see, with the exception of the one leguminous plant, the one that can fix its own nitrogen, soy, they all show a consistent decline in protein levels. This is a much larger meta-analysis done by Irakli Lalaz. And what he was doing is, I think, something like 2,000 different observations for minerals. And you can see that the change in mean concentration for the carbon actually goes up, but for most of the other elements, it declines. And we see that, again, for protein, with the exception of soybean, it's fairly ubiquitous. We think plants are very fascinating entities. They have a wonderful ability to adapt to their surroundings. And we think that as the atmosphere becomes carbon rich, plants are going to adapt to it by becoming carbon rich. And the problem is that there is an imbalance between carbon coming from the atmosphere and all the other elements that are coming from the soil. This isn't just for crops, but also for beef. This is some work by Augustine et al., 2018. This is looking at the growth of cattle under two different conditions. The little a and the AC and the AT are ambient carbon dioxide and ambient temperature. This is ambient carbon and elevated temperature, and you can see there's no effect there in terms of percent nitrogen over a 7-year average. But if I increase CO2, whether the increase is at ambient temperature or elevated temperature, there is a drop in percent nitrogen in the protein content, and the cows put on less weight. They don't grow as quickly. Well, let's go beyond that. Let's add the little bit of the OMG part. What if it isn't just people food? What if it's more than that? Let's look at bees. Now, bees have two sources of food. One is nectar. That's their carbs, right? But the other one is protein, and that's their pollen. So our bees need 10 essential amino acids, and they get that from their protein from the pollen. Is CO2 affecting that pollen? Is the change that's already occurred affecting that pollen? How do we determine that? Well, we decided to give a call to the Smithsonian Natural History Museum. And it's a really cool place if you've never been. You know, everything from dinosaurs to elephants to the Hope Diamond. But way in the back, right to the right of the Ark of the Covenant, you'll find, okay, I made that part up. You'll find all these old gray metal cabinets with plant samples. And the plant we chose was goldenrod. Goldenrod is one of the last flowering sources of pollen for bees before they overwinter. So we wanted to use this historical data as a means to try and assess what the recent change in carbon dioxide has done with respect to protein concentration of this pollen source. So we did this through our CHN analyzer. We spent a lot of time in the bowels of the Smithsonian cutting up what we hoped were pollen and anthers and other parts. You know, your eyes get a little tired after a while. And then we also wanted to do some experimental evidence. And this is an experiment that is at the USDA by Wayne Polly. And it's really cool. What he's done is along the plains, along the prairie here, he has, let's see if I get my arrow to show up. He has these sort of Conestoga wagons. And as you go from here out to the distance, you're going from high CO2 at one end, and as the plants photosynthesize, they're drawing CO2 down so that by the time you get to the other end, you're at preambient levels. And fortunately, he had enough solidago growing along these transects that we'd actually look at the pollen from these transects as a means to do it not only historically, but also experimentally. And here's some of the data. This is the data from the Smithsonian. These are not individual data points. I think there's something like 20, average of 20 based on the year that they were sampled. But there's the estimated protein based on the nitrogen levels, which we used as a proxy for protein. And found that essentially the change in the estimated protein was about 25 to 30% since the 1850s, 1860s. Our oldest sample was 1846, I think, from just outside the Alamo in Texas. Yeah, that's pretty cool. And then looking at the carbon and nitrogen ratios, which have also gone up accordingly. And this is the data from Texas, the experimental data showing the percent protein, different numbers, bigger standard error curves because we have fewer samples, but again, the same sort of relationship that as CO2 rises, the protein concentration is declining. This is in Proceedings for All Society. So let's go and take a deeper dive into nutrition. Let's begin with rice. This is a FACE system, a free air CO2 enrichment system in Japan, in Tsukuba, and you can see it's very different than the one I showed you earlier for loblolly pine. But it does pretty much the same thing, except this time we wanted to look at a lot of different cultivars to see how much variation there were between different cultivars of rice. And this is percent protein for 18 different lines of rice. Some of these are japonica lines, some of them are indica lines, because our second site was in southeastern China, which also has a FACE system for rice. And basically what we found was that there is variation there, and that's a good thing. Because from a breeding perspective, then you might be able to move forward and find these lines that are not showing a decline. But it's fairly clear that there's a ubiquitous effect in terms of rising CO2 on protein. Also, with respect to minerals, there were a number of lines that showed a significant decline in iron and zinc, which also have obviously implications with respect to the human diet. And then we were, for some of the lines, the ones in China, able to do a vitamin B analysis. I don't have the significance up there, but cumulatively they all showed a significant decline. And essentially B1, B2, B5, B9 were all significantly affected by the increase in CO2 that was from the FACE system. And I think in the FACE for both sites, it averaged around 550 to 560 parts per million, so not much further into the future. And interestingly enough, alpha tocopherol, vitamin E, went up. And we were just like, you know, our mouths were agape. How can everything else be going down and this one going up? What's going on? So we put together a working hypothesis, what we like to call a firm possibility of a definite maybe. And what we came up with was if you looked at the secondary chemical, and if the molecular weight of that had a lot of nitrogen relative to carbon, so vitamin B9, for example, 22% of the chemical formula of vitamin B9 is nitrogen. It's very high in nitrogen. Whereas tocopherol has no nitrogen in it whatsoever. That the more nitrogen it had, the more it was adversely affected. If it had no nitrogen, it was positively affected. And we think related to this carbon nitrogen imbalance between what's happening in the atmosphere and what's happening in the soil. Again, I'm not saying this is a law. I'm not saying it's a theory. It's a firm possibility of a definite maybe. We need to do more work to see if this is true. In fact, we just finished a study looking at caffeine in coffee. Now, caffeine is a bicyclic alkaloid that has a lot of nitrogen in it. And we found that for Robusta lines, they just, CO2 kicks the crap out of them. The caffeine drops by half. But for some of the Arabica lines, it depends on the line. Some of them did, some of them didn't. We're still scratching our heads trying to figure that out. So there you go. There's also an income aspect to this, I think. One of the aspects was to look at those countries where at least 50% of the calories come specifically from rice. So that, I think that we came up with 10 countries, Laos, Indonesia, Bangladesh, and so forth. And we looked at their GDP and then related the protein deficit to GDP and the mineral deficit to GDP. And what we found was that the more money you make, the less impact this will have on you, in part because of the diversity of diet. Bangladesh was one of the worst ones, in part 70% of their calories come from rice. They don't have a high GDP. That's pretty much where they get their food from. But as your monetary value increases, you buy more things, your diet diversifies. And this is the vitamin E and vitamin B aspects of that as well. One of the things I'm working on currently is the poverty and food security for the 10 poorest countries. This is the production in terms of staple crops. This is the population. This is the food production in million metric tons. And you can see that production is starting to fall away, even though population is not. And to do this in a slightly different way, here's the kilograms per person per year. So population is a function of the intake of calories, and you can see this is starting to fall off. So we think that in context, and this is, again, the sort of 6-ton T-Rex in the room is food security in the context of food production and climate change. So we think that when we talk about that, my sense is, and this is just my sense, is that the economics of this have to be included, because I think it's not going to be an equal vulnerability across the board. I think that those that are the least, the poorest countries are going to be the ones most affected. And then finally, here's taking the CO2 effects on protein. Irakli Lalaz did this for me. For cassava, maize, potatoes, rice, sorghum, and sweet potatoes, which are the main staples for these countries that are poor. And you can see that in addition to production, it's also going to have an effect in terms of protein concentration as well. Yeah. So in terms of the OMG factor, we see that there are a lot of things here that we just don't understand, that there are going to be interactions, but we don't have a good sense of that yet. It's going to interact by multiple means in human health, from air quality to medicine, to narcotics, to nutrition, and so forth. But it's more than that. Plants are the basis of all life. If they're affected by climate and CO2, what's the impact going to be? Let's say, for the sake of argument, that the bee paper is indicative. What insects do as the amount of protein declines in the environment? What does that mean in terms of the entire food chain? And we don't have a good sense of that. One of the things that strikes me as a plant person, this little bit down here, this is the sum of all animal biomass. Humans are about 0.06 gigatons carbon. All the animals are about 2 gigatons carbon. Plants are 450, and you're affecting how they grow. How many of you have taken more than one plant class? You're missing out on 99% of biology. And I think understanding what carbon dioxide and climate mean for plant biology. Dr. Nachman talked a little bit about some of that, but it's so critical, it's so important, and we don't really have a good sense yet of what is happening. So hopefully, if nothing else, I've given you a 5-cent guided tour of some of the science behind this, and when I could go into depth on some of the issues. But there's so much more we need to know, because it affects every aspect of our lives. Right. So I'm hoping that we can begin to acknowledge these interactions, that there is potential for basic and applied work that we can begin together for both, academics and private industry to come together to begin to look at that and to see where we can find common ground, and if possible, begin to address some of these issues. I think they're really critical. So anyway, thank you very much for your time. Yes.
From the perspective of food security, what do you think are the best adaptive strategies on a global scale that we can do, given that CO2 is only increasing?
I think one of the best things we can do, twofold. One is diversity in what we grow and intensification in what we grow. One of the things that concerns me is that for many of the poorest countries, they've been able to keep pace with population through extensification. They simply plant more, but the yield per acre has not changed. That only changes through intensification, where you make an effort to breed, or you make an effort to add more fertilizer, or you make an effort to manage more effectively. So I think that diversifying in terms of the number of varieties that we have, but also focusing on intensification as opposed to extensification, I think are keys. But, I can't solve that in 2 minutes. It's more complex. Yes, Sam.
Thank you so much. So you mentioned the soybeans, like they seem like less effective in
Yes.
terms of the protein. And I
Yes.
was wondering, like I am a big fan of lentils and legumes
Yes.
in general. So are these also similar to the soybean?
Yes, I didn't have a chance to show, there's not a lot of work with lentils per se, but most of the work has been on soybean and peanut. And peanut also doesn't show a change in protein. It tends to be high. That being said, we did see some changes in protein allergens.
Mm-hmm.
We want to know more about how CO2 is affecting that.
And then it was, I was wondering if, as you were talking, if carbon was the only thing that went up because everything went down. And then you showed the example for vitamin E. And then I know you talk about arsenic, and here we are, the arsenic superfund research program. So maybe you can tell us a little bit more, like this nitrogen thing for arsenic. Does it mean arsenic will go down, up? Should arsenic go down?
We did an initial study with the Arabidopsis. Eaton was our person who worked with us on that, and found that with additional CO2, because of the effects of CO2 on transpiration, CO2 tends to close the stomates of the leaves so that less transpiration goes through. We saw less arsenic in the plants. That's a lab study with different Arabidopsis genotypes, so I'm not sure how much we can move that forward. So that may be a benefit to that. But in terms of the flooding, we have these areas where additional flooding is occurring, and the effect that has on the geochemistry of the soils in terms of making more arsenic available. That's something that I think we should need to dig into a little deeper and find out more about if possible. Do you have a question?
Yeah, I was wondering, were any of the plants you looked at genetically modified?
The soybean was. I have to go back and look. The rice, no. The GMO rice is proprietary, and so we weren't able to get a hold of any of that. Well, I make an exception. We had one hybrid line for rice in those 18 lines. It also showed a decline in protein. So, yes.
So based on jumping off of Ron's question here, so could one make a GMO crop that is less sort of respiratory efficient to like decrease the growth, but sort of increase the nutrient content?
Sure. We think I was, in terms of the variability with respect to protein in rice, there were a couple that didn't show that decline. And so that offers some opportunity there to look at those. But there's also a sort of a silver lining to all this, if you will. If CO2 is plant food, why aren't we breeding for the best rice varieties that are going to take more of that CO2 out of the air and make more rice? Make more rice that is the same amount of nitrogen in it. And who looked up the marijuana? If the marijuana breeders can do this, why can't the rice breeders? Why can't the soybean breeders? Why can't the barley breeders. But it's a very much from a economic point of view, what has happened now with the food system, it's a little, I'm gonna get a little wonky here, but they rely heavily on sort of an assembly line approach where they want uniformity and everything. So they don't want diversity. So if I go to McDonald's and I order fries, which I like, sorry, but they're all from russet Burbank potatoes, right? So if I order, if I get a chicken, it's one of 2 varieties of chicken that are currently grown. Any food that you get now is very genetically regulated. And the problem is that as climate changes, it helps to have greater genetic diversity, not less genetic diversity. So that puts developed countries' food chains, I think, under greater stress to some extent. And that doesn't get talked about very much. Does that make sense? Okay. Yes.
You briefly mentioned the study where you tracked glyphosate resistance with greater CO2 in one species of plant.
Yes.
Is that something you expect to see across multiple species, or do you think it will be more on a case by case?
No, we've seen it on a number of different species, and I think last time I checked, and it's been over a year, so I'm probably out of date, but there are at least 12 different weed species that were showing increased resistance with more CO2 to glyphosate. And we recently published a study working with a Brazilian scientist and a scientist at the University of Arkansas. One of the things that's happening because of the overuse of glyphosate is obviously resistance is increasing very rapidly. And so one of the questions we had is, if I have a resistant biotype, weed biotype, and a sensitive biotype, is one of those favored with more CO2 compared to the other? If CO2 is a selection factor. And so we looked at jungle rice, which is a common weed in rice cultivation, and found that it was in fact the resistant line that responded more to carbon dioxide than the sensitive line. So we think this has a number of ramifications, including the other data that I just touched on, with respect to pesticide usage. And another aspect of that is temperature. And I was talking to you, I think, a little bit earlier today. We think that frost is a major constraint on pest pressures in agriculture. If you have a frost, your number of biological pests diminishes greatly to just a few. And what we found was going from a transect of GMO soybean, because it's all pretty much the same GMO soybean now that's grown in the US, 95% of all your soy is GMO. Going from Minnesota to Louisiana, looking at average yields per acre, soybean didn't change, which surprised us because we thought, you know, the warmer environment in Louisiana, they must have greater pest pressures, and so the yields must go down. But they didn't. And we shook our heads and couldn't figure that out until we looked at pesticide usage. And then there's this logarithmic increase in pesticide usage beyond a certain temperature. The temperature varies depending on whether it's an insecticide or herbicide or fungicide, but they all shoot up dramatically once you reach a certain critical threshold. And we think that threshold in turn represents the sort of biological limit of the pest. With respect to, at least with respect to soybean, I suspect it to be different depending on the crop. Okay, thank you all very much. Are there any questions from online?
No.